Today let us discuss the physiology of digestion. The process of digestion. Now what is digestion? Digestion is a process where complex food substances are converted into simple food substances. So this is a complex substance, these are simple substances. The food we take, the balanced food we take, includes carbohydrates, proteins, fats, vitamins, minerals, water, and fiber. So the balanced diet that we take in includes all these substances. The carbohydrates. The carbohydrates include starch, all forms of starch, rice, roti, cereal grains, whichever cereal grains we are taking, they are in the form of carbohydrates. They are all carbohydrates which are in polysaccharides form, complex form polysaccharides. Proteins. Now carbohydrates, they give their daily fuel. Today I am doing some work. For the amount of work that I am doing today, I require some energy. So the daily energy I get from the carbohydrate sources. Proteins. Proteins are building blocks. They are useful for repair of a damaged tissue. In our body, some cells they die daily and they are regenerated. Proteins are building blocks. They are useful for growth. In children, they are useful for growth. They are useful for repair of damaged tissue. After operation, after accident, when a tissue is damaged, so that tissue is resensitized with the help of proteins. For the formation of antibodies, they are useful in immune system, immunity. All antibodies are synthesized from proteins. Fats, they are reserve food. Under certain conditions, they are oxidized to produce energy. But basically, fat is a reserve food. So these three are in complex form. The other things, vitamins, organic substances, which are required in very, very small quantities. But they have got vital functions in body. Some vitamins are water soluble, some are fat soluble. Water soluble includes B complex and vitamin C. Fat soluble includes A, D, E, K, vitamins. Minerals. There are minerals which are required in larger quantity. Some minerals are required in small quantity. So macro minerals and micro minerals. Each mineral has got multiple functions in our body. Water. It is medium of life. It is the major constituent of plasma of blood. Water is the major constituent of cytoplasm, so it becomes important medium of life inside the cell. All digestive enzymes are hydrolases. When I say hydrolases, they are the enzymes which work in the presence of water. And fiber. When I say fiber, it means when I take a plant diet, substances in that plant diet which are not digested, like cellulose, hemicellulose, lignin, some substances which are not digested, which are in the fibrous form. So fibers, they facilitate clean bowel movements, regular bowel movements, so that there is no constipation. Of course, fibers also reduce cholesterol, they reduce the uptake of glucose from intestine into body. So it's good for diabetics. if you take fibrous rich diet, you can gradually bring down fat. It is good definitely for normal bowel movement, avoiding constipation. But other uses of fiber is it reduces cholesterol. It forms a barrier between glucose and the wall of intestine so that glucose cannot penetrate the wall. So these three, the first three are in complex form. When you take them, they are in complex form. Water, minerals, vitamins, they are absorbed as such. They are already in simpler form, they are absorbed directly. Certain vitamins, they undergo diffusion. Certain vitamins are synthesized by bacteria present in colon, large intestine. Certain bacteria, gut flora, synthesize certain vitamins. So they are absorbed in large intestine. Minerals and water, they are absorbed by simple diffusion. So they are simply absorbed directly into the blood. But these three substances, carbohydrates, proteins, fats, these are in complex form. Now, they are not, they are in complex form. They are so complex that diffusing through the wall of the gut becomes difficult. I mean through intestine. They are non-diffusible. As such, the complex food materials are converted into simpler substances. And in that form, they are absorbed. So conversion of complex substances into simpler substances. So since these are non-diffusible and these are diffusible, that total process is called as diffusion. The conversion of non-diffusible complex organic substances into simple diffusible organic substances which are readily absorbed through gut wall is the process of digestion. Now, when I say complex substances, carbohydrates are in the form of polysaccharides, proteins, they are long chain of amino acids, fats are formed by a combination of fatty acids and glycerol. So they are all in complex form. They are to be broken into simpler form. Now, the act of digestion includes the mechanical digestion and chemical digestion. Two things. Mechanical digestion. Mechanical digestion means in the mouth cavity, with the help of tooth, the food materials are broken down. Inside the stomach, the muscles present inside the wall of the stomach and also inside the wall of intestine. So when they contract, particularly inside the stomach, the muscles when they contract, they cause churning movements of the food. So that is called mechanical digestion. Even in small intestine, the muscularis externa, inside the wall of intestine, there are muscles. The muscles are called muscularis externa. Of course, the smooth muscles, when they are contracting, movement of food, peristalsis occur. So when it is contracting, the food along with enzymes are also mixed. So mechanical digestion is because of muscles present inside the wall. Mechanical digestion also occurs inside the mouth with the help of the teeth, which are useful for biting the food into pieces. Now we will start the act of digestion inside the mouth. Digestion inside mouth. Continue, just. Now, food which is taken into mouth cavity or buccal cavity. Now, the tooth are breaking that food into pieces. Now, salivary glands pour saliva into mouth cavity. We already saw the composition of saliva. చాలా దూరంలో ఉన్నాను నేను. ఎక్కడి నుంచి స్టార్ట్ అవుతాను నేను? Now when saliva is poured into mouth, saliva, it is working at pH of around 6 to 7, 6.8. That is the pH of saliva. Now we saw the composition of saliva. It contains mucus, water. So with the help of that mucus and water, when the tooth are biting the food into pieces, the tongue is helping the mixing of food with that water and mucus to finally form food bolus. Now inside the saliva there is IgA and lysozyme. There is histatin, lactoferrin, lactoperoxidase. There are so many substances which are antibacterial. Lysozyme is present. Lysozyme present in saliva is antibacterial. So it breaks down the cell wall of bacteria. So it's antibacterial. Histatin, lactoferrin, lactoperoxidase, all other substances are also antimicrobial substances. And there is only one enzyme. Of course, that enzyme is called salivary amylase. Salivary amylase, it is previously called as ptyalin. P silent, so we call it as ptyalin. It is salivary amylase. All amylases acts on carbohydrates. The carbohydrate that we are taking generally is starch. The starch is converted into maltose. So this happens at pH 7, at pH 6.8. There is pancreatic amylase. So pancreatic amylase is working above 7 in alkaline medium. So this is working in acidic medium slightly. Inside mouth, there is also one more enzyme called as lingual lipase. But lingual lipase does not have any significant action there. See, all lipases will act only after emulsification of fats. Emulsification of fats occur after bile salts are working on the food. So though there is lingual lipase, there is gastric lipase that enzymes action is not of much significance. So salivary glands have poured saliva into mouth cavity. The tooth have bitten that food into pieces. Saliva has been mixed with that food particles. Salivary amylase present inside, it is converting that polysaccharides into disaccharide. Remember, this is a polysaccharide, this is a disaccharide. Several units polysaccharide. So it's 2 units combined together. 2 units of glucose combined together, it is called as maltose. It can be called as salivary amylase, previously called ptyalin. It is also called alpha amylase, all same. So 2 units of glucose is maltose. However, 30% of starch only is digested in that. And if you are taking food in a hurried fashion, further less digestion occurs inside the mouth. Now, the food mixed with the saliva, it contains water and mucin, so it becomes a food bolus. The food bolus is swallowed. It enters from buccal cavity into pharynx, and when we swallow it, the act of swallowing is called deglutition. It is called deglutition. So food then enters into esophagus. Now digestion in stomach. Now, after deglutition, food has come into esophagus. By act of peristalsis from esophagus, food has finally come into stomach. Remember, generally no digestion occurs inside esophagus. Esophagus is simply a tube-like structure which conveys the food from pharynx to stomach. Now, food has come into stomach. The food stays inside the stomach for 4 to 5 hours. Of course, the length of the stay depends upon the amount of the food that we are taking and the type of the food that we are taking. Difficult to digest substances, they are there for a longer duration. Easy to digest food materials, if they are taken, they are there for a smaller duration. But generally, when food is taken, so it is there for 4 to 5 hours inside the stomach. Now, inside the stomach, mechanical digestion and chemical digestion both occurs. Mechanical digestion means inside the wall of the stomach we have got muscles. I mean the smooth muscles, involuntary muscles. It includes the longitudinal, circular and oblique muscles. They are undergoing vigorous contraction inside the wall of the stomach. Now, when this is undergoing contraction, so you can see some mechanical digestion occurs. But we have gastric glands. We saw the composition of gastric glands. Gastric glands present in cardiac stomach and pyloric stomach, they generally produce mucus. But the glands which are gastric glands present in the fundus and the body, so that area, the glands, they produce various enzymes. Now, enzymes present in gastric juice includes pepsinogen, prorenin, and there is also hydrochloric acid, HCl. Now, we are aware of the fact that gastric juice, the pH of gastric juice is 0.9 to 1.8. So, it's anywhere between 1 and 3.5. So, it's highly acidic. So, hydrochloric acid is present. So, it kills bacteria. It kills bacteria. The major function, though we take cooked food, some quantity of bacteria are still entering along with food and water we take. So, the bacteria kills. So, the bacteria is killed by hydrochloric acid. Now, two enzymes. One important enzyme is pepsinogen. It is an inactive enzyme. A gen at the ending or pro at the beginning indicates that enzyme is generally inactive. Most of the enzymes, when they are formed inside the cells, they are in the form of proenzymes, indicating that they are inactive as long as they are in the cell. And after they are released outside, they have their own mechanism in which that enzyme is activated. So, in which that proenzymes become enzymes. Now, if the proenzymes are present in enzyme state itself inside the cell, it will cause autolysis of the cell. So, to prevent that from happening, generally inside the cell where they are produced, they are in the form of proenzymes. But after they are released into the cavity of gut, they are activated by their own mechanism. Pepsinogen, which is inactive, is activated with the help of hydrochloric acid, HCl. HCl activates pepsinogen. So, it results in formation of pepsin, which is active. Pepsin is also formed by autocatalysis. See, for example, this is pepsinogen. This pepsinogen at the beginning. Hydrochloric acid is here. Hydrochloric acid has come and converted a part of that into pepsin. It started to act, it converted a small part of that into pepsin. Now, this pepsin will act on the remaining pepsinogen. It will act on the remaining pepsinogen, and the remaining pepsinogen is converted into pepsin. So, that's how you can see pepsin which is formed this pepsin converts remaining pepsinogen to pepsin. So this act is called as autocatalysis. So you can see HCl killing bacteria, HCl activating pepsinogen to pepsin, and pepsin formed will undergo, pepsin will help, the pepsin initially formed will help the remaining pepsinogen to become pepsin, so that act is called as autocatalysis. So pepsin is formed by activity of HCl as well as by autocatalysis. Now, the pepsin formed, the pepsin is a proteolytic enzyme. Now, if I observe the proteins, now when I see at the time of digestion of proteins, I told you complex substances are broken down into simple substances. Now, proteins are broken down into proteoses, broken into peptones, polypeptides, tripeptides, dipeptides, and amino acids. See, sequential process of digestion in proteins. The larger protein are broken into proteoses. Proteoses are smaller than proteins. The proteoses are broken down into peptones. That means peptones are further smaller than proteoses. Peptones are broken down into polypeptides, a chain of amino acids. It is further broken down into tripeptides. Tripeptides where there are only 3 amino acids. And broken down into 2, so dipeptides includes only 2 amino acids. And finally broken down into amino acids. We know protein is a chain of amino acids. So it is a chain of amino acids like that. Protein is a huge chain of amino acids. See, these are individual amino acids. When I say dipeptide, 2 of them are combined like that. When I say tripeptide, 3 of them are combined like that. Many of them are combined polypeptide. So peptones are much bigger than polypeptides, and proteoses are bigger than peptides. Proteoses are bigger than peptones, and ultimately the larger one is called proteins. Now, during the act of digestion, you can see a sequential breakdown of proteins to amino acids. Now, the pepsin, the activated pepsin, acts on the first one, the proteins, and converts that into proteoses and peptones. So this area, proteins are converted into proteoses, proteoses are converted into peptones. So this total process occurs with the help of pepsin inside the stomach. Now there is another enzyme inside the stomach. The enzyme is called as prorenin. It's an inactive enzyme. Pro at the beginning indicates that that enzyme is inactive on synthesis. It's an enzyme present in infants. Now HCl activates that to renin. Hydrochloric acid converts prorenin to renin. This renin then The renin comes and acts on casein. Casein is a milk protein in infants, in the suckling infants who are taking in milk. The milk contains casein, a protein called casein. So renin acts on casein in presence of calcium ions. So in presence of calcium ions, it is converted into calcium paracaseinate. It is converted into calcium paracaseinate. Now the pepsin comes, the same pepsin comes and acts on this. So that means it is still a protein. So pepsin comes and acts on that. Acts on calcium paracaseinate to form peptones. So now this is what is happening inside the stomach. Now inside the stomach, the food is staying for 4 to 5 hours. Mechanical and chemical digestion occurs inside the stomach. Mechanical digestion occurs by the churning action of smooth muscles present inside the wall of the stomach. When food enters into the wall of the stomach, gastric gland produces gastric juice. Gastric juice is highly acidic, so its pH is around 0.9 to 1.8. Under highly acidic conditions, pepsin and renin are working on proteins. So these are the enzymes which work in acidic medium. When you go to small intestine, the enzymes present in pancreatic juice and intestinal juice, they are working under alkaline medium. Alkaline medium is a medium above the pH 7. So here conditions are different, there conditions are different. Here condition is that there is hydrochloric acid, so there is a highly acidic medium inside the stomach. Now inside the wall of the stomach, I told you there is a layer of mucus which is 2 millimeter in thickness, which forms a barrier between the acid present inside the cavity and the wall of the stomach. And of course there are also bicarbonates. Now certain cells inside the wall of the stomach are producing bicarbonates. That bicarbonates will neutralize the acid that is produced inside the stomach. The main function of hydrochloric acid is it kills the microbes. The function of HCl is to activate pepsin and activate renin. 2 enzymes are activated with the help of HCl. Pepsinogen activated to pepsin with the help of hydrochloric acid. Pepsin is also formed by autocatalysis. Now that pepsin acts on proteins and converts that into proteoses and peptones. HCl also activates prorenin to renin. Prorenin is inactive and renin is active. Prorenin is inactive. Renin is active. Now HCl activates pepsinogen to pepsin. HCl also activates prorenin to renin. That's the function of HCl. Now after pepsin is formed by activation of pepsinogen by HCl, that pepsin is also formed by autocatalysis. Now that pepsin acts on proteins and converts proteins into peptones and proteoses. HCl also comes and converts prorenin to renin. Renin is an enzyme which is active in infants. Now in infants they are taking in milk as a medium of nutrition. To digest that milk, now in milk there is casein, milk protein. Renin works on that in presence of calcium and converts that into calcium paracaseinate. After calcium paracaseinate is formed, pepsin comes and acts on that, and calcium paracaseinate is also converted into peptones. Now partial digestion has occurred inside the wall of the stomach, inside the cavity of the stomach. Now partially digested acidic food that is formed inside the stomach is called as chyme. It is called chyme. It's called chyme. Now the chyme which is formed will gradually enter into small intestine. Of course there is pyloric sphincter there. Pyloric sphincter regulates Small quantities of food do enter into small intestine. Further digestion occurs inside small intestine.